Hello, everyone. I can see that a lot of users are joining today for this single cell exploration club, so welcome. Any new members that are joining as well, I'm glad to see everyone here. So for today's session, I'll be covering the single cell, well, the spatial transcriptomics exploration club for today. So let me just share my screen so you're able to see that as well. Okay, and just to remind everyone, at the end of the session, normally we do try to circulate a poll where we can gather some questions from the users joining the sessions if the session is useful for them. So I'll share the screen now. Okay. Okay, so for the sort of session of today, what we wanted to really focus on was showcasing really how you're able to mine spatial transcriptomics data, especially if you're working in a context of sort of respiratory system disorders, asthma, or sort of fibrotic tissue or lung tissue. So we really wanted to show users how they're able to utilize SpatialX to be able to analyze both bulk spatial or single cell spatial transcriptomics data sets within the SpatialX system and sort of specific types of questions that they can answer and sort of functionalities that they can utilize to really interrogate the tissue structure, the architecture, both at the micro level, but also looking at sort of distances between different immune cell types and how these sort of cell types communicate with each other. So that was sort of the focus of today. And let me just make sure that you're able to see the full screen. Okay. So I'll go to the next. So mainly what we'll be sort of reviewing is the SpatialX module within the biotrain ecosystem. If you're really new to the team and you're not sure where to sort of identify or find that module, when you sign in into the biotrain ecosystem, you can go directly to the SpatialX module. So this is sort of our multi-image integration platform that really allows you to sort of interrogate spatial, publicly available data as well as your own imaging data. And it does support many different technologies, so it's not just limited to like Visium HD, but you can also look at Xenium data, Cosmics, Vision Merscope data, and other different sort of spatial data technologies as well. So if you're more interested in H&E staining and running image segmentation, then that would be sort of the module that you can use as well. Okay. So just to go or recap before I go into this case study, in the last session, if you weren't able to join the last exploration club, we did sort of a session relating to our B-Browser X Pro module, and we looked at specifically a breast cancer data set to really understand cells that were hypoxic, so sort of tumor cells that were hypoxic based on the expression of ADM and really looking at whether that specific phenotype of those tumor cells had some specific differentiated expression, sort of functional pathways that were relevant within that specific cluster. We tried to perform gene set enrichment analysis to really dive deeper into these tumor cells with the expression of this ADM target that really is more related to like hypoxia adapted cells. And we tried to really uncover what sort of pathways were involved within these specific cells. For example, we looked at immune signaling, starvation response pathways that we can see here in the pathway enrichment analysis. We were able to really look at and distinguish these tumor cells based on sort of specific metacentric metastasis markers, periphery specific markers, and also markers that are more related to the primary tumor site. So there's a lot of different features that you can see here that we're able to perform not only on spatial data, but also single cell RNA-seq data sets that we can dive deeper into very specific cancer cell profiles that might have like a circulatory function, a hypoxic function, or a proliferative function as well. So this is just a recap of that session. So if you're interested in going through these different sessions that we've covered, we do record them. So they're on our sort of YouTube channel that you're al always able to sort of review back on. Okay, so the agenda for today, as I mentioned, will mainly focus on SpatialX. We'll try to dive deeper into what we would term as spatial heterogeneity. So how can we define specific cell populations in severe asthma? And our objectives of this case study will be broken down into three different sections. One which is really looking at how can we spatially map and annotate these pro-inflammatory hubs within the tissue compartments and within just asthma severity. So how can we sort of subcluster specific samples and really look at these hubs and sort of the transcript location. The second one is really having to do with sort of how can we quantify cell to cell distances and define inflammatory or microarchitecture dynamics. So there's a lot of different features that I'll try to sort of show you how you can interact with the image, both by looking at sort of the distances of the different cells. You can run sort of gene query analysis and other sort of functionalities relating to cell distance. And then the third sort of objective was really to look at how can we identify specific therapy resistant spatial niches. So can we subcluster a specific cell population that we were able to determine had this pro-inflammatory hub expression, and can we only look at specific samples and then run differential gene expression to really sort of uncover other markers within specific treatments. Okay. So just before I go into the data set, I just want to give some background on sort of what are the samples that we'll be analyzing or what is the disease context in this case. So really, if we look at severe asthma as a disease model, this sort of model or this disease remains really heterogeneous and this is really a treatment-resistant condition affecting 10% of sort of asthma patients worldwide. And of course, if we're trying to really understand how these biologics work, we can see that within sort of severe asthma, we have many cytokines that are sort of enriched within these cell populations. For example, we sort of see heightened IL-4, IL-5, and IL-13 expression within the tissue. And really what we are trying to understand is how does this affect or how do these different expression values benefit how patients exhibit these persistent symptoms, how they have this difference in structural airway remodeling, and how can we look at sort of non-type 2 inflammatory profiles, looking at sort of using spatial imaging data. So really severe asthma is really characterized by both sort of epithelial integrity as well as activities within stromal, endothelial compartments, and also how they interact with other immune cell types like fibroblasts, macrophages, or other cell types that you might see, as well as mast cells, which are quite important. So really, we'll try to sort of uncover these different localized immune stromal interactions and some of the features or functionalities that you can use to do that. So I think one important thing to mention is that there are a lot of challenges when it comes to analyzing spatial heterogeneity across different tissues or sort of tissue layers, especially in the context of severe asthma. So one of the first ones that comes often across studies is that even after corticosteroid or some form of treatment, there's always this pro-inflammatory remodeling cell hubs that you normally see this sort of higher percentage of mast cells or specific fibroblast clusters that have very specific profiling or specific target expression. And these persist in certain tissue compartments and they vary across different samples or different patients as well. So something that would be really interesting is how can we look at sample variability, so patient variability, and can we see this consistency or is it only specific to certain patients that are receiving certain treatments? So that's something that we can try to visualize as well using SpatialX. The second one is really how these zones may harbor other types of inflammatory cells. So not only sort of the extracellular matrix restructuring signals, but how does this sort of affect inhaled drugs or exposure to these? And the third is really how can we measure these cell-to-cell distances? So there's a lot of different I guess, functions and different ways that we can do this. You can do sort of distance query, you can do cell-to-cell interaction analysis, or look at sort of neighborhood densities as well. So that's a feature that we'll try to cover, as well as how can we look at diameters if we're interested in distance within specific niches that are showing these higher gradients of IL-33 or other growth factors or chemokines in the tissue. So again, these are some of the challenges that a lot of researchers face, especially if they're only working with single cell RNA-seq, but they really want to understand the spatial um, context or the tissue layer context, then you would sort of need to utilize these spatial transcriptomics data sets. So the study that I wanted to sort of focus on was this newly curated data set in our Talk to Data system. So if you do have an interest actually in exploring this data set, since it is new and we've just recently added this study, what you can do is go into the ecosystem and you can go to the request management page and just add a new request so that you're able to access this new data set in the system. So our team will be able to send you that early access link to the study, and you can then explore it even further in SpatialX for your own specific questions that you might have. Okay. So just a bit of context on this study. So this study was sort of performing not only Xenium sort of sequencing, but also had images relating to geomics. And really what they tried to understand is sort of a spatial atlas of both health and healthy sort of control samples and asthma lung tissue. And they really tried to look at the pathologic cellular hubs. So really looking at mast cell axes or fibroblast clusters that might emerge within severe asthma. And they tried to answer very specific questions like, are there any inflammatory hubs that we were able to identify? Are there any tissue compartments that we can see where we see mucus glands and sort of surface epithelium enrichment? And can we look at these sort of signaling gradients across these different tissues? So that's something that the sort of authors tried to uncover. But again, there's a lot of other functionalities that we can utilize on how to quantify proximity of these cells, neighborhood structure, and really looking at spatial gene expression across these different gradients. Okay, so now if we go to the live demo, one of the first questions that we'll try to look at is how do these spatial relationships between immune or stromal cells change across asthma severity. So if we go into the data set in SpatialX, this is where we have all of our different samples for this specific study. So we can actually look at the the combined data set with all the different samples, both sort of severe and healthy control samples, or we can look at individual samples individually. So if you only want to look at asthma severity, you can turn on that sample data and look at the layout display of that sample. But for now, this is all of the different samples that we have in this study. So we can actually look at the actual image of the tissue. We can look at the cell types that are annotated within each of the samples. And really where is sort of this abundance of very specific populations like sialylated cells, or we can look at fibroblast cells that we can see clearly that they're abundant in very specific regions of the tissue. And some ways to really interact with the actual image is that you need to make sure when you're analyzing data in SpatialX that you have your specific preferences turned on or how you're actually visualizing the data. So that means if you're looking at sort of a stroke view of the cells, what is the point size? So you're able to see the actual sort of abundance of specific cell populations in those regions. You can minimize that to actually look at the actual staining of the image. So there's also a staining underneath that might be sort of antibody staining. So you can actually correlate that with the cell boundary. Or you can again change the point size or the opacity so you're able to see just the cell type abundance on the tissue. Okay, so there are a lot of different ways that you can start playing with these sort of configurations, these inputs, so that you're able to really look at those regions or those niches that you're focusing on. You have to make sure that all of the information is turned on, like the antibody staining underneath the sequencing, the cell boundary information or the segmentation, which is really how the actual cell is segmented within the actual tissue structure. So this segmentation is coming from the author directly. So it's already a specific file that we can input. But if there is a case where you want to run on your own data set another segmentation model to be able to compare those differences, we do have that ability where you can either input your own segmentation modeling file, so you want to actually overlay that new segmentation, or you can run our sort of CellPose Cyto3 segmentation model as well. So you can really compare these different cell boundaries and these distances in the tissue. So that's where you can sort of look at this information. Now, if we're trying to really look at not only the cell types, but we want to actually run gene query to identify these niches, you can input those antibody genes here, and you can look at the expression of certain genes on the tissue. So you can actually see where IL-3 is mainly expressed or where it's mainly enriched. And we can see also what sort of cell types are expressing IL-33. So that's a way that you're able to either zoom out or zoom in into very specific samples and interact with the image. You can look at another specific target as well. Or you can look at all genes and really see where is the enrichment of these chemokines and these sort of specific inflammatory targets. So if we zoom out, it makes it easier to actually look at all these different samples. And we can actually subcluster if we're only focusing on severe disease. This is where you can input your metadata category. So you can select what condition you're focusing on. Let's say if we're focusing only on the severe asthma samples and we don't really want to look at the healthy control now, we can select the group and subcluster that specific samples within SpatialX. So now when we query the genes, we can actually see the enrichment profile related to severe asthma only. And it makes it easier to really look at these specific niches where we have the expression abundance. We can overlay the annotation as well. So we're able to see, okay, what are these cell types that are expressing these gene? For example, these are serous cells that actually show really high enrichment of our specific targets. And we can see across these different samples as well. So we can look at each sample individually. So we see that this specific tissue sample has a lot of enrichment of these specific targets. And we can also overlay again the author cell type so we can see, okay, what are these cell types that are expressing? Is it sialylated cells? Is it fibroblast cells that are actually expressing these genes? So this is how you can start interacting with the actual image. If you're more interested in sort of distance query, so you want to look at the distance of very specific cells, we do have that ability to measure these distances directly from the tissue. So you can actually create a distance and sort of calculate that and sort of look at the expression of certain transcripts. You can also lasso a selection. So when you lasso a specific region of the tissue, that will allow you to not only subcluster. So if I go back and go to the lasso, let's say I'm interested in this specific region here. What I can do is perform distance queries. So that will allow me to perform sort of what is the distance between these different immune cell types, and especially in areas where we see sort of this higher proportion of these genes that have sort of a either a remodeling effect or an inflammatory or just chemokines that are sort of enriched within this region. So these are, again, different types of features that really try to understand spatial proximity, distance query, and really how these different cells are located within the actual tissue space. Okay, so I'll just remove or I'll exit this specific group here, and I'll remove this, and then I'll go back again to my condition so I can only locate the asthma samples. Okay. So let's say our next question, if we go back into our slide here, was really to understand what are the changes between immune cells and stromal cells. So if we want to look at actual quantifiable changes, again, you can look at the expression value of the chemokine or the specific target within the image, but there's also ways that you can visualize the data as well. So if we want to look at the expression of these specific genes on the tissue, we can go and create different charts depending on what the question you're trying to answer. So is it proportion specific changes that you're trying to really look at, like what are the changes across different treatments? Then you can create a cell proportion chart. And this will allow you, since we subclustered the healthy control, where it's basically not showing these cells. So what we can do is just remove the healthy control and just show the severe. asthma, and we want to look at what are the different proportions across these different treatments. So this is one way that you can start looking at these different visualizations. You can create a heat map as well to look at these changes. Or what you can do is also create a different type of visualization, which is a bubble heat map that really tries to look at what sort of cell types are expressing these specific chemokines and these targets. So within the basic chart, you can go and select the bubble heat map view. And we can look at not the condition because we know that we've subclustered these specific severe asthma patients, but we can actually look at the cell types within the tissue and how these different markers are differentiated or sort of differentially expressed within these specific cell types. So that's another sort of visualization that you can generate from the tissue. You can look at different sampling sites. So if your main focus is looking at these different regions in the tissue that you might already have either created from region segmentation or from other features or pathology specific regions, that you're able to select those specific sampling sites so that you can see where that specific gene is actually expressed and how that relates to specific cell type populations. So we're able to see here the specific ECM circulatory genes that are mainly expressed by these globulate cells. We can see some genes are also expressed by like the extracellular matrix region. And then we also have other immune cell types that are also expressing these other sort of CCL5 and serous cells that show the enrichment of other specific chemokine targets as well. So again, this is data that you're able to access. And when you hover within the actual node region for that specific cell type cluster within that gene, it gives you a bit more information on the mean expression within that group and what is the percentage of cells, so this percentage of serous cells that are actually expressing this target within just severe asthma because we've subclustered only those samples. So again, these are different questions that, again, you can try to answer to really look at these differences. You can look at sample variation. So a lot of the questions I get quite often is I do want to make sure that it's not only related to one sample or one patient, but can we see this consistency across different patient samples? So if you're more interested in answering that type of question, then again, you can go into the cell proportion and just select that you want to see this variation not across treatments per se, but you want to see it across the actual sample ID. So across these different asthma severe patients and look at if it's actually consistent across the patients or is it only something that we see in certain patients that received very specific treatments. So again, you can also generate these type of tools. You can customize the figure so that you're able to add any changes to that. Or you can export the actual data, so the matrix data from the analysis that you're performing. Okay. So this was more for our first question, just to really get an understanding on the different features that we can perform and how we're able to really look at that expression value across severe asthma. Now, the second question is really looking at cellular neighborhood analysis. So really, how do cellular neighborhoods reorganize? And can we focus that on specific therapy resistant regions? So again, if we go into the spatial image here, what we can do is select a specific cell type group or specific I guess, regions that have an expression of a target. So if there is a case where you're interested in only applying only regions that have a higher expression of those specific chemokines or those targets, what you can do is annotate those regions across the samples. So what you can do is then look at region segmentation only on those regions that you labeled with the expression of a certain gene set. So that's one way that you can do it, or you can just run it on all the samples so that you can see what sort of regions do we have. So in that case, we do have that feature within SpatialX, the region segmentation. And it's a bit different. So I do want to make that clear that it's not based on the actual image segmentation. What it's trying to do, it's trying to look at spatially distinct regions that might contribute some biological understanding. So it might have regions that have this co-localization of certain genes, signature markers that might be relevant to treatment response. And it tries to sort of look at the spatial pairwise gene correlation correlation analysis within these different regions in the tissue. So that's really what it's trying to calculate and trying to delineate these distinct biological functions by doing this pairwise gene correlation analysis in the database. So if we go into region, I can show you here, you can add some information on what you're trying to run. So if you're focusing on certain samples, you can subcluster them before you run region segmentation. And then currently we have the spatial gene correlation analysis, but we are planning to add other types of methods that you can sort of perform this type of analysis. Advanced parameters are more related through thresholds that you can add as well. So that might be specific to the tissue that you're working with. So you can adjust those thresholds as well for that methodology. So if we go into the sort of analysis here, we can zoom in a bit closer to the severe asthma samples that I subclustered, and we can look at this region segmentation result. So we can see that within these different samples, we are able to really define 5 distinct segments within the samples, and we can actually zoom in to look at these different segments as well. And we can look at also what are the actual gene sets that are enriched within these segments. So we're able to really sort of define these different segments. We can look at the AU-cell score of these gene sets directly on the tissue and the sample so that we're able to actually later on label. So if we're able to define, okay, this segment 1 has a very specific expression of these epithelial stem-like zone factors, then I can actually lasso that region and then annotate it with that specific gene set. So it really gives you some context on this sort of spatial region segmentation, and it gives you the AU cell enrichment score as well. So if you want to actually look at a higher enrichment region, you can just select by doing the distribution and looking at those specific regions that have this higher AU cell score. Within each of these segments, let's say we're interested in looking at this specific one that has a higher enrichment of CXCL6 or L33. We can go into the pathway feature within the region segmentation and actually go deeper into what are the pathways that are involved within these regions in these samples. So for example, we can see that there's an alpha beta integrin signaling pathway that are really showing the enrichment of these genes that are relevant or located or localized within our specific sample. And we can also see other specific pathways that are sort of mediated cell adhesion that are quite important as well in asthma, or other specific modules that might give us some indication of what type of regions are we really looking at, and is it sort of a hyper-inflammatory region? Is it one that there's some remodeling effect, or some that might be specific fibroblast region as well? So again, this is another type of, I guess, functionality that you can run within the system that really gives you that context on these sort of pathway and functional regions in the sample. Okay, so if we go back into our slides, let's say our third question was really looking at neighborhood analysis or neighborhood niches. And we might want to look at what is the composition across different spatial domains, and can we see these different gene correlation regions, or can we get more information on that? So for that type of feature, we can actually run the neighborhood analysis. So it's not cell-to-cell communication per se. We're not looking at ligand binding interactions, but what we're really trying to understand is the co-localization of certain sort of groups, and we're trying to really look at if this is specific to certain treatments. So if we're planning to look at only specific drug treatments, you can also subcluster those regions from the sample, so that you're only focusing on regions that are treated with a certain sort of treatment target, and you can then run the neighborhood analysis to see if there's any cell type co-occurrence only within those regions that are based on that treatment. So if we zoom in into the spatial tissue, we can look at the actual cell type co-occurrence. So it tries to really calculate the correlation score across these different cell types and how they're co-occurring across the tissue space. And then the more important part is how can we look at neighborhood clustering analysis within the sample. So this is another way that we can start really looking at what is the proportions of these neighborhood cells within the tissue. The more, I guess, the lower you go within the actual intensity heat map, the more granularity you'll see on the actual samples. So you'll see a lot of different spatial domains. And we can look at that through a stack bar. So we can see, okay, we have these different spatial domains. So what is the proportions of these different sort of neighborhood cells within the tissue? And the more sort of granular, I guess if you don't want it to be that granular, you can go into the heat map view. And you can look at lesser proportions, so you're able to see just the larger proportion of cell types and how they're neighboring within those spatial domains. So again, that's quite important, especially if you're subclustering only a specific treatment site, so specific samples that were treated with a specific agent, and you want to look at the spatial domains and how these different neighborhood cells are clustering within that domain. So again, this is another type of analysis that you can perform. You're always able to save either the metadata that is sort of created from the neighborhood analysis or the actual data and proportion information between these different spatial domains. So again, this is another feature that is quite important, and it gives you more understanding on that type of analysis. And again, just to recap how you're able to select different treatments in case you are planning to only look at let's say just the effect of imatinib or another type of agent, then you can subcluster only cells that are relating to that treatment before doing any neighborhood analysis. Okay. So again, if I go back, I can go into our slides just to see if there's any specific things that we missed or what did we cover today. So I guess some of the summaries from this session was really an exploratory thing that I wanted to show users how to get acquainted to working with spatial imaging data in the SpatialX system. So the first, I think, question that we covered was really how can we quantify these different immune and stromal hub structures within the actual image. So again, we covered that you can use distance query. So that's when you're specifically lassoing a region in the tissue that has the enrichment of, like, let's say a gene that you're looking at, and you want to run distance query on that specific region. You can look at gene query as well, which really tries to understand the expression of specific targets within regions, or you can even subcluster very specific cell type populations and look at that further in the tissue. The second part that we also covered was really look at neighborhood and proximity analysis. So that's using the neighborhood analysis function and really trying to detect not really ligand binding specific interactions, but just sort of how there's proximity or the proportion of different neighborhood cells within these different spatial boundary regions, and really how that can be distinguished between different treatments. So you can actually subcluster different treatment groups and then run the neighborhood proximity analysis on those regions specifically. And again, the last one was really having to do with how are we able to run region segmentation. So really pinpointing specific expression of chemokines or alarmin genes or remodeling genes, and really understanding if this is a specific fibroblast cluster. Can this be targeted? Can we look at a specific enrichment score of these gene set within these different regions that are identified? So again, these were sort of the questions I wanted to answer, and I wanted to show all of the researchers that joined today so that they're able to use it for their own data or even explore this similar type of data set in the system. Okay, and I think now that our team can send the poll just to get an understanding on your feedback or sort of questions that you might have on the system, I do want to highlight that there are a lot of features in our pipeline that we're developing in SpatialX, and some of them we've actually released very recently. So we do want to get some feedback from your team on how the deconvolution works. So if you're working on bulk spatial data sets like Visium, Visium HD, and you're not able to run like our cell type prediction algorithm that is mainly based on like the single cell resolution that is segmented on the tissue, you can do deconvolution, and we do want to get that feedback on that specific function. So if you're interested in running that function, you'll see that turned on when you upload any Visium or any bulk spatial data set. You would be able to run the deconvolution path feature. So it's currently turned off on my data set because it's not a bulk spatial data set, it's a Xenium data set. Okay. There's other features as well that we do want to get some feedback. So if you can try the region segmentation that we covered today or the neighborhood analysis, and we will be releasing in the next coming week the cell-to-cell interaction functionality. So if you're also interested in going deeper into what are the ligand binding interactions that are occurring within specific cell types or specific spatial domains, then that's another functionality that you'll be able to run, and also pathway enrichment analysis that can be based on a specific pathway or a GEO specific pathway list. So that's another feature that we'll be releasing very soon. So again, we want you to sort of utilize SpatialX, and we want to gather your feedback as well on these. Okay, so I guess for now, since I do want to leave some time for users to fill out the actual poll and the quiz that we left or we circulated. If there is any questions from the team that they want answered, you can add it in the chat, and we can follow up with you as well if you have very specific questions on how to access the data. So I can see here that we have, can we import and export the Seurat object? So for this SpatialX system, yes. So if you're planning to analyze or create new metadata from any study that you're analyzing, you can either export the metadata from that study, or you'll have an option here where you can actually export the object of the study. So when you're creating a new export, it's going to ask you some information on what you're trying to export from the data, and then it'll export directly on your local computer. Okay. And if you're able to export the Seurat object of a public data set, then we can follow up on that as well, so that we can sort of explain how that works in the talk to data system. For the region segmentation, can we visualize cell type enrichment? So yes, that's actually something that we're going to be releasing very soon. So yes, in the region segmentation, when you perform that analysis, it's really a model that it doesn't let you select the genes that you're inputting. So it mainly maps out these functional specific regions based on those gene sets that are highly enriched. So you are able to see the actual AU cell score within each of the samples, and you can sort of gate that specific score or looking at those pathways within that segment. If you're interested in using your own pathway, then that's going to be available in the pathway analysis feature that we'll be releasing very soon. So you would be able to select what specific pathway you're actually trying to look at and what are the genes as well that you're trying to run within the system. Okay. So how does the region segmentation differ from neighborhood analysis? So I guess to clarify that a bit further, so the region segmentation, what it's trying to do, it does like a pairwise gene correlation analysis to be able to identify specific segments in the tissue that are enriched for a specific gene set population. So it gives you some context if there is some inflammatory regions or some, let's say, not pro-inflammatory, but some remodeling specific areas that are based on ECMs or extracellular matrix remodeling or specific targets. So that's more of a functional pathway enrichment that is trying to map out different spatial domains based on that region functional segmentation. For the neighborhood analysis, it's a bit different. It's more related to what are the proportions of these neighboring cells and do they differ across these different spatial domains? So it's more related to that sort of cell proximity interaction, and you get these proportion data sort of matrices that you can actually export to look at these different proportions in the image. So I hope that made that a bit clearer. If you do have any questions and you want to understand those features a bit more, what you can do is go into the tutorial, into our documentation, and that gives you the ability to go through our documentation, and we actually go a bit deeper into the functionalities themselves and how we actually calculate that information in the system. Okay. So thank you so much for the questions. I'm very happy that you were able to join today, and I hope this was useful so that you're able to use it on your own data as well. So we look forward to seeing you on our next week's session, which is more on our Asia time zone, but we will be having another sort of EST time zone webinar as well. Okay, so thank you very much, everyone.